Welcome everyone. This is Dr. Shruti Analyx, Assistant Professor, Department of Biomedical Engineering, and today we'll be looking at the topic regeneration of nails and lens regeneration from the Unit-4 Ectoderm-Derived Organs, which is taken in Tissue Engineering and Regenerative Medicine. So first coming to the anatomy of nail. The nail is composed basically of a hard keratin protein, which is very tough, and it helps to protect the fingertips, and it is produced from the epidermal cells. The nail plate is the visible part of your nail, which actually grows out from the matrix region. So from the matrix, the nail region is growing. So the matrix is basically the growth center of the nail. The living tissue basically produces new keratinized cells that eventually form the hard nail plate. And lunula, which is the half moon shaped area at the base of the nail plate, it is the visible portion of the distal nail matrix. And cuticle is the thin layer of dead skin cells that grow out from the eponchium and which adhere to the nail plate to form the protective seal. And there is a onychodermal band, which is a thin grayish or glass-like band, which is located where the nail bend ends. This is the free end basically. And the pulp is the fleshy part of the fingertip. So because this fingertip is rich in a lot of nerves and blood vessels, to protect this tip, the nail is being provided, or to help in supporting and to protect it, the nail is being given. Nail generally has a fast healing ability, and it has a high cell turnover in the matrix. It ensures that there is quick recovery after injury. Let's say you break a nail, or you cut your nails, there is generally the nail grows back very fast. It depends on person to person, age, etc., but there is generally good growth in the nail. The blood supply and the nerve endings in the nail bed generally support for this good repair. Next, coming to the growth and repair mechanisms, we have some important stem cells. So first is the nail matrix stem cells. So the nail matrix stem cell, which is the LRG6+. This drives the continuous regeneration of the nails. And there is wind signaling that happens in the nail, and the wind signaling basically helps to support the stem cell activation and for the nail formation. And there is BMP signaling, bone morphogenetic protein signaling, which drives the differentiation of the nail cells or nail stem cell LRG6+ into the proximal nail matrix, which harden into the keratin producing oncocyte. So the oncocyte are the mature nail cells which will form the nail plate. So from the LRG6+, you have the proximal nail matrix, which then mature into the keratin producing oncocyte, and this produces the keratin plate or the nail plate, and that is the visible part of your nail. If you are looking at the nail plate, RG6+ stem cells are at the base of your nail or the fingertip, and as they differentiate, you have the proximal nail matrix, and then the further away it has the keratin producing oncocyte, and these keratin producing oncocyte is what is producing the nail on the top. It is getting all the nutrition from the nearby blood vessels. The increased level of the LRG6+ proliferation in response to the growth cues helps in driving the nail plate generation. So when there is high amount of the LRG6 plus activation, there is more amount of nail growth that happens. Next, we'll be looking at lens regeneration. So the lens epithelial cells are the main cells that are involved in the lens regeneration of your eyes. So these are generally found under the anterior lens capsule, and they proliferate and differentiate into new lens fibers. So if we are looking at the eye, so if you're looking at the lens, the or the more proliferative cells are towards the equator or the ends of the lens. So at the end of the lens, more of your stem cells are present, and as they differentiate, the more differentiated cells are appearing towards the center of the lens nucleus. So initially you have the stem cells, and from the lens stem cells, and from there the progenitor cells are coming, and from there you have the posterior subcapsular cells that are forming, and as they differentiate, they form the lens cortex, which are more arranged, and then the lens nucleus cells. So around 30,000 cells are produced in this cycle every year. The anatomy of the lens is very peculiar in nature, and we need to understand that this is maintained by a group of stem cells. So this stem cells, as we see here, um, you have multiple levels of stem cells that are finally forming the lens nucleus, which is transparent at the center. But the stem cells that are found at the equator region, they are like normal cells, but only after they proliferate and differentiate, they are becoming the lens nucleus, which is showing this transparency. As we move towards the center, which is the lens nucleus, the cells become more and more transparent in nature. So we would wonder, how can cells become transparent? Because all around your body we have cells, and they generally have a specific color, but how in specific in the nucleus the cells are very transparent in nature? So this is due to the a few reasons. So first, coming to the organelles. So the lens cells, which are towards the center of the nucleus, they do not have any organelles. So as they differentiate and come towards the center, the mature fibers lose the nuclei. So these lens nucleus fibers, they will lose their nuclei, their mitochondria, the endoplasmic reticulum, and thereby, because there are no organelles inside the cells, they will eliminate the light scattering. Also, they do have certain specific proteins, which is the dense crystalline proteins. These are packed at very high concentrations, which will create a uniform refractive index within the cells. So the presence of proteins is another reason, crystalline proteins. And then the precise cell packing. These lens nuclei are usually packed in a specific manner, so they are usually tight and orderly packed, and they are hexagonally arranged, and this minimizes the presence of any intercellular gaps. And because of the lesser intercellular gap and also the minimal extracellular space, the the refractive index interfaces are reduced in general, and that helps to maintain the transparency of the lens nucleus. And so the reduced refractive interfaces due to the lesser intercellular space and the extracellular space, helps to make sure that there is lesser bending or scattering of the light. So thereby the lens nucleus is transparent, whereas the cells towards the periphery are not. Okay, so it is only the center of the lens that maintains its transparency, and this transparency is very important because as the people age, the formation of opaqueness that can reduce the visibility of the person. So this, the regeneration process is very critical for the proper vision of all people. So next, coming to the key signaling pathways that are involved in the lens regeneration. So the first one is the PAX6 molecule. So this is a master transcription factor or a master regulator of the lens epithelial cell proliferation and differentiation. So this activates the lens cells proliferation, and it initiates the fiber differentiation by turning on the crystalline gene production. Okay, so the next is the fibroblast growth factor or the FGF. This FGF drives the lens epithelial cell migration, elongation, and survival. So, this FGF stimulates the lens epithelial cell growth and fiber formation. So, the gradient of FGF concentration determines the fiber maturation zones that are present along the lens. Next is the BMP or the bone morphogenetic protein. This works along with the FGF to control the lens regeneration. So what it does is it will modulate the FGF activity, and it fine tunes the differentiation pace and prevents the premature maturation and maintains the LEC progenitor pool in general. So this is the function of the bone morphogenetic protein. So I'll be ending this lecture note on a clinical insight. So usually, let's say when a cataract surgery is done, the cataract surgery leaves behind the lens epithelial cells. Some can even regenerate the lens-like tissue, but the problem with this is that the lens cells that are usually formed are often disordered. I had told you that the proper packing is very important for minimizing the refractive interfaces, and that is important for the transparency. But when we are trying to regenerate the lens tissue from the uh, existing lens epithelial cells, what happens is we are left with disordered cells, and because it if the cells are disordered, we do not get good transparency. Another strategy that can be done is that we can look at these signaling pathways. So lens epithelial cells regeneration can be done to restore with vision without artificial lenses. So we can look at activating one of these signaling pathways, and thereby PAX6, or we can work with increasing the fibroblast growth factor, and thereby help in the proper or improved lens formation. So to improve the lens regeneration, we can look at these kind of growth factors, or another strategy is we can look at tissue scaffolds. So scaffolds that are either natural or synthetic, that can support the proper formation of the lens cells. So in this case, we need to take care that the scaffold that is used has to be transparent, and it does not hinder the transparency property of the lens stem cells. With this, we are going to end the this session of our lecture. Thank you.